get started with our last session of the symposium for this year. This is kind of like our medical health session, so I'm excited to hear a lot of these talks. Robert Waters will be with us today talking about Staph aureus and Pseudomonas, so I will turn it right over to him.
Thank you. Hi guys, like she said, I'm Robert Waters, and I've been working with Dr. Carty, Dr. Keller, and Dr. Austin over at LeadCom, and we have been exploring the effect of preventing and treating Pseudomonas and Staph aureus with cinnamaldehyde ointment. So today we'll go over what cinnamaldehyde is. I'm sure not everybody here is a microbiologist, so we'll go through these two different bacteria. We'll talk about how we made the ointment, and then we'll talk about the results we got from that and our conclusions due to the results. So to start out with, we'll look at Staph aureus. What is Staph aureus? As you can see up there in the top right picture, it's a gram-positive cocci. It can cause a variety of issues, but what we're mainly concerned with, since we're looking at ointments, is skin infections. So stuff like impetigo, folliculitis, things of that nature. And the example of folliculitis down here, where you're seeing infections of the hair follicles leading to inflammation and a lot of bacteria sitting there. So one of the issues with Staph aureus, which I'm sure all of you have heard of MRSA, with MRSA they have what is called the MEK-A gene. With that, they're able to make different penicillin binding proteins that no longer the antibiotics no longer recognize because they're structurally different, just enough so the antibiotic can't see it or interact with it. And they also make penicillinases, which is an enzyme to help degrade different types of bacteria, different types of antibiotics as well. So over time, it's becoming harder to treat. These are just two quick examples of why it's becoming hard to treat them. The other bacteria we're talking about today is Pseudomonas aeruginosa, and as you can see up there on the top right, it's a gram-negative rod. And once again, we're more concerned about skin infections. And mainly you only see Pseudomonas infections in immunocompromised patients because it's an opportunistic pathogen. It's everywhere though. It's probably all over this room. Sometimes you even survive through disinfectants. It's pretty crazy. But like I said, immunocompromised patients, so diabetic foot wounds, burn victims, you might see them on those as well. Over here is an extreme example. So here you got a burn victim, and that green tint, the green pigment, is actually due to Pseudomonas growth. So Pseudomonas, when it grows, actually produces the green pigment. So all that green back here is Pseudomonas on that guy's back. So once again, with gram-negatives though, it's intrinsically harder to treat them because they have stuff such as efflux pumps in their outer membrane, which, let's say the antibiotic gets into the cell. The cell's like, I don't want you here, shoots it right back out. So it has no time to actually function and stop the cell from living. Then also it has decreased permeability. So they have something called porins also in the outer membrane. Well, it can select for what can actually come into the cell. So if it doesn't want the antibiotic to come into the cell, it just doesn't let it in. And lastly, just like Staph, they have the potential to degrade antibiotics. So both of these bacteria, one, they cause skin infections, and two, over time they're becoming harder to treat. So we're trying to look for a different treatment approach or a different prevention approach to taking care of these bacteria other than antibiotics. So before we jump into cinnamaldehyde, we need to take a step back and actually talk about essential plant oils. So basically what essential plant oil is, it's basically a natural compound that you find in the plant's leaves, the bark, the roots, different parts of the plants, and basically take it and you go through steam distillation and you produce the oil from it. And a lot of times these oils have nice fragrant smells to them, so you'll see them in cosmetics such as perfumes, even soaps and detergents. I'm sure you've had stuff that smells like eucalyptus, orange, lemon, stuff of that nature, what comes from the oil from those plants. And interestingly enough, these oils have been shown to have antimicrobial effects, and not only that, they've been shown to have a broad spectrum antimicrobial effects. So not only can they stop the growth of Staph aureus, a gram-positive, but they can also do the same thing with Pseudomonas, which is a gram-negative, like I said before. So why cinnamaldehyde? So we, over the last year or two, we've tested up to 40 different oils against Staph aureus and Pseudomonas, and we found that cinnamon bark oil especially had the greatest effect against Staph aureus and Pseudomonas. So after we saw that, we looked into it a little further and found that about 80% of cinnamon bark oil is actually made up of one compound called cinnamaldehyde. So we decided to take that, just one compound, and compare different percentages of that compound to the cinnamon bark oil as a whole. And when we did that, as you can see right here, CBO indicating cinnamon bark oil, CA is indicating cinnamaldehyde, we found that there was no significant difference between 100%, 75%, and 50%, as well as 25%, when compared back to cinnamon bark oil. And what we were really happy about is the 100%, 75% maybe had a difference of maybe 1 or 2 millimeters as far as the zone, which I'll get into in a second here what that is. So when we saw that, we're like, you know what, we're not going to deal with all these other components in this oil. Especially, one of the issues with essential plant oils is there's no really big regulation to them. So you can call it a cinnamon bark oil if it has somewhere between 65 to 85% of cinnamaldehyde. So other than having to deal with that potential confounding variable every time, we decided to use cinnamaldehyde by itself, so we have to deal with different percentages between batches if we use that. So the way we prepared cinnamaldehyde ointment is that we actually found an article that was written by Seen et al. In 2014, where they're actually using, I believe it was eucalyptus ointment to take care of fungi and look at fungi and see how it affected fungi. And so we took that same recipe with PEG 400, PEG 4000, and water, and made a prep with 2% cinnamaldehyde with preservative, which we use as methylparaben, and without preservative. And then we make controls for both of those without the cinnamaldehyde as well. And you see kind of a little example here of the ointment. It's kind of hard to see. It's white, so it's kind of hard to see on this background. So the way we tested this is actually we did what is called a cell or a well diffusion method. So we had 3 strains of Pseudomonas and 3 strains of Staph aureus, and we'd streak it across the plate. And then we'd take this little hollow cylinder here, it had a 6mm diameter, and we push it right into the agar, make a well, and pull that piece of agar out with it. And then we would add either the 100% cinnamaldehyde or the ointments directly into that hole. And we incubate it overnight, come in the next day, and measure what is called a zone of inhibition, or a ZOI. So what a ZOI is, it's actually an area where the oil has actually diffused out and prevented the growth in that location. So as you can see here, both these plates are with the 2% cinnamaldehyde ointment with the preservative. One on the left is Staph, and the one on the right, which is a green collar, is Pseudomonas. This area from here to here, all that clear space, that's where the ointment actually diffused out and prevented the growth from occurring in those locations. And then over here, same way with this little clear area right here, ZOI right there as well. As expected, we saw Staph had a lot larger zones than with Pseudomonas, which is what we usually see with all the oils we tested. But when we actually looked at the data, I only have time to show one strain of each bacteria per experiment, just because of time constraints. But as we saw with all 3 strains of Staph aureus, there might have been a difference about plus or minus 2 or 3 millimeters, but we always saw the exact same trend, where we had, cinnamaldehyde at 100% being the best, followed a little closely by 2% ointment plus paraben, followed by the 2% ointment alone, and there was always a significant difference between the cinnamaldehyde and the 2% ointment. But, interestingly enough, when we looked at our controls down here, which our control was always a 6 because we had that 6mm well, so that's always part of the measurement. So anytime we have a 6, nothing occurred, didn't have any growth inhibition, didn't do anything like that. But what we found here is our paraben with the ointment alone in our control did absolutely nothing. But when it was actually in conjunction with the cinnamaldehyde, we actually might have saw a little bit of a synergistic effect occurring because it had a greater effect with preservative than without the preservative, which was definitely a surprise. I mean, a good surprise, but it was definitely a surprise to us when we saw that. So here's an example of the Pseudomonas. Once again, with Pseudomonas and Staph, for all the strains, we always saw the exact same trend where we have cinnamaldehyde followed by the 2% cinnamaldehyde ointment, with the preservative, followed by the 2% ointment alone. So next, after we saw that, we were able to prevent the growth. We wanted to see if we could essentially treat it. So what we do is pretty similar to what we did with the first method, but in this case, we would take a lot of bacteria on the plate first, incubate that overnight, and then the next day come in and make a well, the 6mm well, and then add the ointment or the cinnamaldehyde. So on the left, once again, before I get into that, once again, they're both with 2% cinnamaldehyde with preservative. On the left, you have Staph aureus. As you can see, there's no zone forming. We were unable to take care of that. We tested, we swabbed the locations multiple different days around where we thought the zone could have been. Bacteria was always there. We grew up in different media, grew up in different plates, it was always there. But, surprisingly enough, when we did Pseudomonas, you can see there's a little different tint there. So we were a little interested, so we decided to swab those as well, put on different media as well, and then put on different plates, and there's no growth. The bacteria there is dead. All that's left over is just a little bit of the pigment after the bacteria has died. So interestingly enough, and I'll get into it more in the conclusion, we were able to essentially treat Pseudomonas with this ointment, but not able to treat Staph. So, like I said before, here's the Staph graph. Anything with a 6 means it had no effect. Didn't cause any kind of growth inhibition, just basically sat there and did nothing. But we saw the exact same trend with all 3 strains of Pseudomonas, where we have cinnamaldehyde followed by the 2% cinnamaldehyde ointment with the preservative, followed by the 2% ointment alone, and then the controls obviously didn't do anything. So, in conclusion, we found that the cinnamaldehyde 100%, when compared to the 2% cinnamaldehyde plus preservative, there was no significant difference between those two, indicating that we could possibly use the 2% cinnamaldehyde ointment as a prevention technique for both Staph aureus and Pseudomonas skin infections, or before they can actually occur. And secondly, the 2% cinnamaldehyde plus preservative could potentially be used as a treatment approach on Pseudomonas infections. Obviously not Staph, but interesting enough with that is, like I said in the beginning, gram-negative is intrinsic hard to treat because, like I said, they have efflux pumps, they have all that stuff in the outer membrane. So we were really surprised when we actually saw Staph aureus, we weren't able to affect that, but Pseudomonas we could. So in the future, what we want to do is actually look at cinnamaldehyde toxicity. Most of cosmetic industry, like anything that has the smell, or anything that has the essential plant oils in them, it's usually about ranges between 2.5% to 5% of the cosmetic is actually made up of essential plant oils. But obviously, to be on the safe side, we need to go through and do toxicity screenings of this to make sure the percent we're actually using at 2%, which we pick to stay lower than what we usually see in cosmetics, is still safe. After that, we want to do, Dr. Carty actually knows someone in Texas that has a mouse burn model, where essentially they will knock out the mouse, they have basically a pre-measured square they put in the back, and they will do a 3rd-degree burn so there's no pain involved. And then we can add the bacteria to that, followed by the ointment, or try to prevent it with the ointment, then add the bacteria to see how that comes out. And lastly, like I've mentioned before, we want to determine why were we able to essentially treat Pseudomonas but not Staph aureus. It's definitely one of those surprising things that we did not expect as well, but it's definitely something we wanted to figure out, maybe by trying other gram-negatives versus other gram-positives, see if we keep seeing the similar results, and then go through and try to figure out what the actual mechanism of action is for that ointment. All right. That's it for me, guys. Are there any questions?
I know that using essential oils, especially if they're like just pure, there's often like a risk or some kind of like warning, that can cause irritation to the skin. Do you have any idea with burn victims, with it being at 2%, if they didn't experience any discomfort, or you think that's probably? That's
something we'd have to definitely test. We don't know that yet. So we're trying to get it down in dilution as far as we can. Obviously, 100% is going to burn your skin, so we're trying to get it diluted down as much as we can. As far as how that would affect burn patients, I don't know. We're not that far yet. Definitely want to try that out. But yeah, we're obviously not that far yet. Yep.
Essential oils are real popular right now. Will there be more studies in the medical field?
There's actually quite a bit of studies now. There's a lot of people trying different bacteria, different essential plant oils. It's just something we definitely got interested in. Dr. Carty actually did it, I think, part of her PhD thesis. And so we just kind of extended onto that and just kept going with it. But yeah, I think a lot of stuff is going toward a direction. A lot of people are looking at that direction because it is definitely could potentially be a nice alternative approach to using antibiotics if we could counteract it. And also, on top of that, in my master's thesis, we actually found that when we combine these cinnamon bark oil with an antibiotic that doesn't function anymore against Pseudomonas, we were actually able to get it to work again. So we actually had a synergistic effect between the two at really small concentrations. So there's also that approach as well, potentially.
I have a quick question.
Oh, yeah.
Why aren't they regulated? Why are essential oils
It's just,
not in the...
I mean, it's just one of those things where it's just standard to have somewhere between 65 or say % of cinnamaldehyde to 80%, and they go, oh, that's fine, it's all cinnamon bark oil. It's just they find the main component essentially, or the components in there, and they say you have to have this percentage, but it's a very wide percentage, and obviously it can lead to a lot of compounding variables if we're, one day using one that has 60% cinnamaldehyde, which is the main active ingredient, and next day we have one with 80%, obviously that's going to lead to a potentially big difference between the two. Now, as far as why they aren't more regulated, I mean, I don't know. It's just one of those things that aren't really, it's not really out, they don't really come out and say, hey, this is going to treat all these infections, because they don't have any proof of that yet. So I guess that's why they're not regulated, I guess, because they don't come out saying like, oh, we can treat this now.
Yeah.
So no one's really has them regulated because they're not coming out with those kind of statements, I guess.
That's good that you guys are getting studied then.
Yeah.
Any other questions?
Oh, yep.
It's not so much a question, but is it also because it's really regulated what you can say as far as treating anything?
Yeah.
And the minute you bring in pain, then, the FDA's like, no.
It could possibly be, yeah, because I mean, once you say that, you're kind of throwing yourself out there, and if anybody disproves it, then you're in a lot of hurt then at that point.
Well, thank you very much.
Thanks.
Okay, our next speaker is Yusra Mansour, and she will be talking about auditory midbrain hypoplasia. Yusra?
Okay, so good morning, everyone. Thank you for coming. My name is Yusra Mansour. I'm currently a second-year medical student over at LECOM. I'm going to talk to you about the research I did over the last year investigating changes in the auditory brainstem in an animal model of autism. So I want to start with a little bit of background on autism spectrum disorder. ASD is a developmental disorder that causes troubles with communication and interactions with other people. Now, individuals with ASD exhibit various signs and symptoms, including failing to respond when someone calls their name, although they may respond normally to other noises, and altered sensitivity to sensory inputs like light, touch, smell, and sound. Now, evidence of hearing difficulties in ASD is extensive, so I just want to summarize some of the key findings. The majority of individuals with ASD have difficulties with hearing and understanding speech. They have problems with verbal cues, hypersensitivity to loud noises or hyperacusis, are easily distracted by background noises, and have trouble even localizing sound. Now, studies of the auditory brainstem response show clear evidence of abnormal brainstem processing in individuals with ASD. So I want to quickly talk about the transmission of sound from the outer ear to the inner ear. So your outer ear is essentially shaped like a funnel. So it focuses and filters all of those sound waves hitting the side of your head into your ear canal. Those sound waves then hit the tympanic membrane here and cause it to vibrate, which causes all of the ossicles in the middle ear to vibrate as well. And the last ossicle is called stapes. It's actually the smallest bone in the human body, and it attaches to the cochlea via the oval window. So if you look at the cross section of the cochlea right here, you can see that it's filled with fluid. So the vibrations of the stapes are transmitted into fluid waves in the cochlea. Those fluid waves then depolarize the hair cells in the cochlear duct right here, and then signals are sent back to the brain via the cochlear or the auditory nerve. So the beginning of this slide is essentially what we finished with on the last slide. You have signals being sent from the cochlea to the cochlear nucleus via the auditory nerve. The cochlear nucleus then projects bilaterally to the superior olivary complex, and from there bilaterally to the lateral lemniscus and the inferior colliculus. The inferior colliculus projects again bilaterally to the medial geniculate and the auditory cortex, with most of the projections actually being to the contralateral side. Now, our study focuses mainly on the auditory midbrain, including the nuclei of the lateral lemniscus and the central nucleus of the inferior colliculus. So before we dive into the results and the hypothesis of the study, I think we need to talk a little bit about valproic acid. Valproic acid is an anti-epileptic that's known to cause an increase in the risk and the incidence of autism spectrum disorder. So because of this, in utero VPA is an accepted and proven animal model of ASD. Now, previous studies have shown us that in utero VPA exposure has caused a significant impact on the number of neurons, their morphology, and their structure and function in the lower auditory brainstem. So because of these results, we hypothesized that the auditory midbrain, including the nuclei of the lateral lemniscus and the central nucleus of the inferior colliculus, are also significantly impacted by in utero VPA exposure. So this slide is essentially a visual representation of the timeline we use for VPA exposure. The pregnant females were exposed to the vehicle or peanut butter on embryonic days 7 through 12. Now for the control group, it was just peanut butter on all those days, but the VPA group had VPA added to the peanut butter on embryonic days 10 and 12, and that's represented by these blue bars here. The females were then allowed to give birth normally, and the pups were let go until postnatal day 50, when they were perfused and the brainstems were removed and weighed. Now our data is based on 11 control rats from 5 litters and 8 VPA exposed rats from 3 litters. So if you look at B here, you can see that on postnatal day 50, when those brainstems were weighed, that the brainstems of the VPA group weighed significantly less than those of the control. Okay, so on this slide, we're looking at the central nucleus of the inferior colliculus. A and B are images of the entire inferior colliculus, with the central nucleus outlined and highlighted in yellow. And in B, we could see that there was actually a reduction in the overall size of that central nucleus in the VPA group compared to the control group. C and D are higher magnification images of the neurons in the central nucleus, and we actually found a significant increase in the size of the individual neurons following VPA exposure. And if you look at this neuron here, you can tell that it's larger than these up here in the controls. So A is showing us essentially what we discussed on the previous slide, that there was a significant increase in the size of the neurons in the VPA group. And B is showing us that not only was the average size larger, but the entire range was shifted to the right. And that's consistent throughout the entire central nucleus. The ventral, center, and dorsal aspects of that central nucleus all had significantly larger neurons. Now we also looked at the orientation angle and the shape of those neurons, but we saw no significant change in VPA group compared to the control. But if you take a look at F, although we found that the neurons are larger, there was actually a significant drop in the number of neurons. Now the control inferior colliculus had about 200,000 neurons, and in the VPA group, that number dropped to only about 140,000 neurons. So we also looked at the nuclei of the lateral lemniscus. In yellow here is the dorsal nucleus of the lateral lemniscus, green is the intermediate nucleus of the lateral lemniscus, and blue is the ventral nucleus of the lateral lemniscus. And just like in the central nucleus of the inferior colliculus, we found that the overall size of the nucleus was significantly smaller following VPA exposure, and that's shown in B compared to A. Now the dorsal nucleus of the lateral lemniscus, or the DNLL, is the only nucleus where we found an increase in the size of the neurons, because we found no significant change in the INLL or the VNLL. If you look at these neurons here, you could tell that they're larger than the neurons in the control. So again, A is showing us that significant increase in the size of the individual neurons in the DNLL. And C, D, and E show us that just like in the central nucleus of the inferior colliculus, there was a significant decrease in the number of neurons following VPA exposure. So next we looked at calbindin expression. Calbindin is a calcium binding protein that's expressed in quick-firing neurons. Now, previous studies have shown us that in the superior olivary complex and the cochlear nucleus, there is a significant decrease in the number of calbindin positive neurons following VPA exposure, so we wanted to look at the auditory midbrain. If you look at A here, it's an image of the DNLL, and the DNLL is the only nucleus of the auditory midbrain that consistently expresses calbindin in controls. You can see a lot of these fluorescent green calbindin positive neurons indicated by the arrowheads, and a couple calbindin negative neurons indicated by the white arrow. And if you look at B, it's clear that there's a significant decrease in the number of these calbindin positive neurons, although we did find that the surviving neurons were larger and had significantly smaller axon diameters, and that's shown in C and D here. So next we looked at paraneuronal nets. Paraneuronal nets are networks that develop in an activity-dependent manner. They're thought to stabilize synaptic contact after the onset of hearing. So we thought that since VPA exposure significantly impacted auditory development, we'd see a decrease in the number of these networks. So A and B are images of the inferior colliculus, and they're showing us the distribution of these paraneuronal nets, and that they're actually more concentrated in the ventral medial aspect of this nucleus in both control and VPA exposed brains. In C and D, we're getting closer looks at these networks. The white arrowheads are pointing to the neurons that don't have paraneuronal nets, but the black arrowheads are pointing to those neurons with the white halos around them that are essentially locking synapses in place. Now, unfortunately, we did not find a significant change in the number of paraneuronal nets in any of the nuclei of the lateral lemniscus or in the central nucleus of the inferior colliculus. So we also looked at tyrosine hydroxylase expression. Tyrosine hydroxylase is an enzyme involved in the synthesis of the neurotransmitter dopamine. So we used its expression to determine VPA's effect on the dopaminergic inputs. So we saw in previous studies that the superior olivary complex and the cochlear nucleus had a significant decrease in the number of tyrosine hydroxylase positive axons following VPA exposure. If you look at A and B here, they're images of the central nucleus of the inferior colliculus. And in the control in A, you can see a lot of fluorescent tyrosine hydroxylase positive axons indicated by the arrowheads. If you look at B, it's clear that there's a significant decrease in the number of these axons. The central nucleus of the inferior colliculus is the only nucleus of the auditory midbrain where we found a significant decrease in the number of these axons, and that's shown in C. This leads us to believe that there is a loss in the dopaminergic input following VPA exposure. So this slide essentially ties everything we discussed together. In A, it's showing us the number of neurons in all of the nuclei of the auditory brainstem, and you could really appreciate the size of the central nucleus of the inferior colliculus compared to all of these other nuclei. So in controls, the entire auditory brainstem contained about 226,000 neurons. Now following VPA exposure, this number dropped to only about 179,000 neurons. Now 71% of those neurons lost were actually lost in the central nucleus of the inferior colliculus. And if you add the number of neurons lost in the nuclei of the lateral meniscus, this number jumps to about 86% of all neurons lost in the entire auditory brainstem. Now, since we said that the central nucleus of the inferior colliculus is so massive compared to these other nuclei, we wanted to normalize the number of neurons in the other nuclei to the number of neurons in the central nucleus of the inferior colliculus. Now, when we did this, 4 nuclei stood out with the largest proportional loss, and those are these with the red dots, the MSO, the VNLL, the INLL, and the SPON. All of these nuclei lost at least 43% of their neuronal population. But if you take a look at the 4 nuclei in the red box here, these all have inhibitory inputs to the central nucleus of the inferior colliculus. Now, since 4 of the 6 most affected nuclei are inhibitory, this leads us to believe that there is hyperexcitability in the central nucleus of the inferior colliculus following VPA exposure. Now, just to summarize, we found that in utero VPA exposure caused significant dysmorphology and hypoplasia in the auditory midbrain. And just like we saw on this slide, there was a preferential effect on inhibitory inputs. And we also saw a significant decrease in the expression of calbindin and tyrosine hydroxylase in the auditory midbrain. Question?
I know someone who was on the autism spectrum and also had a brain tumor on the brainstem. Would you propose that it has to do with the already compromised brainstem?
With the already compromised brainstem? So a lot of autism spectrum disorder is during the embryonic days, so I don't know if the brainstem was after or during, but it could have kind of caused the, there's no more balance in inhibitory and excitatory inputs, and that could have caused it.
What is valproic acid prescribed for?
Seizures, migraines, it's usually seizures, but because it has a lot of side effects, other drugs are chosen, but it's usually seizures and migraines, and bipolar disorder.
I have a question. Do they, with this knowledge, do they not prescribe it to pregnant women, or is it the woman doesn't know it's pregnant yet, or she's pregnant yet and is still taking the medicine?
Right, so it used to be prescribed a lot when they didn't know, but now that they know it's teratogenic, other drugs are chosen over this if they know that the female is pregnant, or even if she's just of the age where you could potentially be pregnant.
Oh, okay. Any other questions?
I'm sorry if I missed it, but is there a difference between the number of exposures that they had? If they had more exposures, the effects kind of compound on each other?
Right, so we wanted to control so that the data is usable, because if it's different, it won't. So we used the same amount on those days, but yeah, I'm not sure if I could guess that if there's more, there's probably more impact.
Thank you very much.
Our next speaker is William Alter, and she's going to also continue discussion on the auditory brainstem. And these last 3 talks have all been from LECOM.
Good morning, everyone. So I'm William Alter. I'm a first-year medical student, and I'll be presenting on the impact of neonatal excitotoxicity on the auditory brainstem. So just a quick overview on the auditory pathway. There's a sound. That sound wave will collect in the pinna, or the external ear, and it's going to propagate through this auditory meatus, or auditory canal, causing vibration of the middle ear ossicles, which will eventually lead to displacement of the fluid found within the cochlea, right here. That will eventually lead to activation of the spiral ganglion neurons, causing afferent axonal projections to this cochlear nucleus. This cochlear nucleus will then be activated and cause projections to the superior olivary complex. And those projections will then go to the inferior colliculus via the lateral meniscus, and then eventually to the medial geniculate, and finally the primary auditory cortex. So just a little overview on glutamate. Glutamate is the most abundant excitatory neurotransmitter found in the central nervous system. It is stored and released by both neurons and astrocytes, and is released at the synaptic terminals. So if there's an excess of glutamate, that can cause overstimulation of these AMPA or NMDA receptors, which will cause an influx of calcium. That influx of calcium can cause cytoskeleton degradation, as well as opening of the pores of the mitochondria, which will lead to release of reactive oxygen species, as well as caspases. And that can lead to apoptosis or neuronal cell death. So just the overall purpose and hypothesis of our study, we wanted to research and examine the impact excitotoxicity has on the auditory brainstem. Specifically, we'll be looking at the spiral ganglia, the cochlear nucleus, and the superior olivary complex. So in order to do this, we utilize monosodium glutamate, or MSG, which is a naturally occurring sodium salt. And we use MSG as a way to induce the environment of excitotoxicity. In no way, shape, or form did we use MSG as a way to see how it impacts the auditory system. We used MSG as a way to create this environment of excitotoxicity. So in order to do this, we have the male and female rat together until the female got pregnant. And then on postnatal day 4 through 10, we selected the male rats and injected them with either 4 milligrams per gram of MSG or an equivalent solution of saline. And then on postnatal day 21, the male pups were weaned, and on postnatal day 28, the male rats were perfused via the ascending aorta, their brains were extracted and weighed, and then they were used for immunofluorescence. So as you can see here, after weighing the rat brains on postnatal day 28, you see that there is no significant decrease between the MSG exposed compared to the control, but you do see that there is a trend towards decreasing. So this image right here is just a cross-sectional area of the temporal bone. So this temporal bone is what houses the cochlea, and within the cochlea are these spiral ganglia that we were looking at. And this is an image of the control spiral ganglia. As you can see, these red arrows are pointing to the spiral ganglion neurons. You can see that there are a lot of them. They're dense, they're compact, and you can even see their axonal projections right here. Now, after MSG exposure, you see that there are a lot less neurons there. You can also see that there are fewer axonal projections. So then when we looked at their density, we saw that there was a significant decrease in the density of the spiral ganglion neurons in the MSG exposed compared to the control. But what was interesting was that we saw that there was a significant increase in their cross-sectional area. So this right here indicates that there is compensation occurring in the auditory system after this neonatal injury. So again, we looked at the spiral ganglia, we looked at the cochlear nucleus. So what is the cochlear nucleus? So it is the first auditory brain station where integration of this information occurs. This is also where globular bushy cells are housed. So globular bushy cells are found within this cochlear nucleus, and they receive excitatory input from spiral ganglion neurons. So this right here is just a cross-section of what the cochlear nucleus looks like. So if you look right here, the little doggy ears, that's your cochlear nucleus. It houses those globular bushy cells. And this is what globular bushy cells look like. So they're round with an eccentric nucleus, and they're found between this auditory nerve right there. So we want to quantify the amount of globular bushy cells in the MSG exposed compared to the control. And when we did this, we saw that there's a significant decrease in the number of globular bushy cells in the MSG exposed compared to that of the control. We also want to see how the excitotoxicity has an impact on both excitatory and inhibitory terminals. So again, excitatory terminals come from spiral ganglia to the globular bushy cells. So we'll be using vesicular glutamate transporter, or VGLUT, to be able to see the excitatory terminals. So when the globular bushy cells receive projection from the spiral ganglia, they're known as modified end bulbs. Now, the globular bushy cells also send projections to the superior olivary complex, specifically to the medial nucleus of the trapezoid body, or MNTB. And when it does this, it's known as the calyx of Held, which is right here. So this is one of the largest axons, and it allows for fidelity of the input. And as you can see here, it's so large that it encompasses the cell like so. So just a little recap again. So globular bushy cells project to the medial nucleus of the trapezoid body, or the MNTB, and excites it. When the MNTB gets excited, it sends out glycinergic or inhibitory axons to the lateral superior olive, or the LSO, the medial superior olive, or the MSO, and the superior para-olivary nucleus, or the SPON. So when we looked at this, when we utilize V-glute, we see here that after MSG exposure, this white arrow is indicating a globular Bushy cell, and the yellow surrounding the red cell, the globular Bushy cell, is indicative of excitatory terminals surrounding the cell. You can see that there are far fewer yellow dots surrounding the MSG exposed compared to that in control. And that indicates that there are fewer cell excitatory terminals. And as you can see here, we saw that there was a significant decrease in the number of excitatory terminals surrounding each globular Bushy cell in the MSG exposed compared to the control. Now when we looked at the MNTB, again, MNTB is what sends out the inhibitory terminals to the, or inhibitory axons to the SOC. And it receives one of the largest nerve terminals known as the calyx of Held. When we looked at it, you could see that there is less immunofluorescence in the MSG exposed compared to the control. And when we looked at it, we saw that there was a significant decrease in the number of excitatory terminals surrounding the globular Bushy cells compared to the control. We also wanted to see how the inhibitory terminals were impacted after excitotoxic injury. So we utilized vesicular inhibitory amino acid transporter, or VIAAT, to be able to visualize this. Now, you'll see this expressed in most nuclei. So as you can see here, this is also the cochlear nucleus. Remember the little doggy ears? As you can see in the MSG exposed, in a previous study done in our lab, they did see that there was a significant decrease in the size of the cochlear nucleus, and you can actually see that pretty well here. You can see the MSG exposed one is far more narrower, and you can see that there are fewer cells. And the control is larger and has more cells. But when we looked at the inhibitory terminals surrounding both the MSG exposed and the control, you can also see that there are fewer inhibitory terminals surrounding the MSG exposed compared to the control. These little white dots or magenta dots surrounding them are less here compared to here. And that's exactly what we saw. And we did this also with the MNTB. We also saw a significant decrease in the number of inhibitory terminals surrounding the MNTB compared to the control. We also wanted to look at the superior olivary complex. So the components of the superior olivary complex include the medial nucleus of the trapezoid body, or MNTB, the lateral superior olive, or LSO, the medial superior olive, or MSO, and the superior para-olivary nucleus. So these are very important because they are what allow us to localize sound. So the LSO is important because it allows us to detect high-frequency sound. And the MSO is important because it allows us to detect low-frequency sound. So just to give you a little example of this. So say I clap my hands. That intensity of that sound is going to be detected by my lateral superior olive. So now let's say I clap my hands again. That time it takes for it to reach my left ear versus my right ear is going to be detected by my MSO. So they're really important for localizing sound. So in order to be able to visualize the inhibitory terminals or determine the amount of inhibitory terminals surrounding components of the superior olivary complex, we utilize optical density measurement. So if there's an increase in the optical density measurement, that would quantify with an increase in the number of inhibitory terminals surrounding these cells. And what we saw was that there was a significant decrease in the optical density of the lateral superior olive. So that indicates to us that there is a significant decrease in the number of inhibitory terminals surrounding the lateral superior olive. What was interesting is that we did not see a difference in the MSO or the SPON. So let's just bring this all together. I know that was a lot. So we saw a significant decrease in the density of the spiral ganglion neurons, but an increase in their cross-sectional area. So remember, this is the forefront of this all. This is where our pathway began, our circuitry began. So a disruption here will probably lead to a downstream impact, and that's what we're going to be able to see. So the system is compensating by having an increase in the cross-sectional area, even though there's a decrease in the density. So because the spiral ganglia project afferent axonal projections to the globular Bushy cells, and there's a decrease in the number of spiral ganglia, we should see that there is a decrease in the number of excitatory terminals surrounding globular bushy cells. And because globular bushy cells also project excitatory axons to the contralateral MNTB, and there are fewer globular bushy cells being activated or stimulated, you will see that there was a significant decrease in the number of excitatory terminals surrounding the MNTB as well. So if you recall that MNTB also sends out inhibitory projections to components of the superior olivary complex, specifically the LSO, MSO, and SPON. And we did see a significant decrease in the number of inhibitory terminals surrounding the lateral superior olive. So what does this all tell us? This suggests that the rats there's a potential that they are unable to localize sound. And that is because the circuitry was disrupted from the very start, because we saw the significant decrease in the number of in the density of the spiral ganglion. Any questions?
Why was MSG chosen for this study versus like other limiting things?
So MSG is cheap, and it does cause excitotoxic effects. So because it's utilized and it's readily available, you know, obviously labs all cost money. So we want to make sure we get, you know, our money's worth, right?
I have a question too. So you did this, if there was exposure later in the life, the lifespan, if it was an older individual that was exposed,
Yeah.
do you see the same things or no? Because it was exposure when kind of like the circuitry was forming.
So actually we specifically chose postnatal days 4 through 10 because they are actually more susceptible to injury during that time. But as for humans, there are individuals that experience excitotoxicity. So individuals that have like ischemia, seizures, strokes. So people that go to concerts and they play loud music, and that can cause excitotoxic effects on your ears or your system. Any other questions?
Thank you very much.
All right, our next talk is a speed talk. If you saw in the newspaper just recently, it was only like a week ago, there was a picture of it. Maria is from the Erie County Department of Health, and she'll be talking about frozen dragons puff treats. Very unique title.
Hello, I'm Maria Liggett with the Erie County Health Department, and I'm going to talk to you about the frozen dragon puff treats. The mission statement of the Erie County Department of Health is to preserve, promote, and protect the health, safety, and well-being of the people and the environment of Erie County. So as I'm out doing my inspections, I try to keep safety and protection of the community in mind. So, how many of you know what Dragon Puff snacks are? Well, it is a snack made from Korean cereal, and then it is blasted with liquid nitrogen like this. When ingested, it causes a vapor to form in your mouth, and you can blow it out of your nose or your mouth like smoke coming from a dragon. So in August, I was up at the mall, and this new snack craze tried to come to the Erie Mill Creek Mall. But I brought it to the attention of my director, Karen Tobin, because I was not comfortable with licensing this facility. And I also had to find out what kind of liquid nitrogen they were using. There's a medical grade and a food grade. So I went and I closed them down because they did not have proper licensing procedures in place. And I had to investigate more about the product. So we researched the product and found that they were not using food-grade liquid nitrogen, and that across the country this was making children aspirate and could burn them, as well as people making the Dragon Puff treats. So our team, with help of our director, Melissa Lyons, Mill Creek Township Code Enforcement, Erie County Department of Health physician and medical directors, we came up with this restrictions for selling the product and safety requirements. Some of those were that they needed food-grade liquid nitrogen. They had to have proper safety equipment such as gloves and goggles while using the liquid nitrogen. They had to have a landline phone in case they had to call 911 if someone would aspirate. They could not sell to minors, and they must eat in front of the employee. So we met with the Dragon Puff company and told them that these restrictions and that they would have to follow the requirements. They would have to call Mill Creek Code Enforcement and put up signage like this before they could open. So the health department put together this signage that said this product may cause severe injury, including respiratory issues, damage to internal organs, and skin damage. So the day that the company wanted to open, Mill Creek Code Enforcement would not allow the nitrogen tanks in the Mill Creek Mall because it is a Class H3 hazardous chemical. And the FDA issued a statement advising the public not to consume this product. So the Dragon Puff, the production of Dragon Puff cereal has been frozen from operating in Erie County, thanks to the collaboration of the members of the community that want to keep the residents safe and healthy. Thank you, and are there any questions? And Dave Bruce did do a story on it. Yes?
Yeah, I do have a question. Okay, now, the liquid nitrogen thing and the protocol that you guys put together
Yes.
for this, I mean, I kind of get that.
Uh-huh.
But for instance, like over in Cleveland, where they have to do all this ice cream, okay? And that's prepared with liquid nitrogen into the ice cream. So doesn't stuff like that, without investigating fully a little bit more, make it a little harder for some of these businesses that are trying to bring in more responsible products like that ice cream?
Well, that would be a totally different case altogether, because what they actually did, as I go, we can go back, well, even in that picture there, they put the cereal, which is like little balls of Trix or something, and then they spray it with the liquid nitrogen right on to the balls, and so that is very cold. And also a liquid can form in the bottom of the cup where they could also drink it. When they're making ice cream, it's a whole different procedure. It's not actually, injected and then you eat it right away.
My worry is more like for later on, like regulations later, like it's like you already established a precedent and it just makes it harder for business, that's all.
Well, I don't know so much as a president because we're just trying to keep people safe and healthy. And this was a whole different, like when I first closed them down, when I went up there and they were already operating without a license,
No, I get that.
they did not have food grade liquid nitrogen. So they were using tanks of liquid nitrogen that could have had any chemicals that we had no idea what was in there. So just protect the public. Is there any other questions? Yes.
Some of the other countries that have had this or other states, like have we been in operation for like a number of years, or is it something that's like relatively new and novel?
Yeah, I think it's a new craze that just happened this year. They did have problems with it down in Florida and like at fairs and festivals where the children were getting burned or aspirating because they couldn't breathe from the cold temperatures. So the FDA has been researching it and they are advising people not to consume it. Any other questions? Thank you, Mary.
Our next speaker, Dominique Baker. She's from LECOM, and her talk is a speed talk as well, and she'll be talking more about essential oils. Dominique?
Good morning. My name is Dominique Baker, and I'm a second-year medical student at Lake Erie College of Osteopathic Medicine. And my experiment, the comparison of antibiotics and essential oil preparations on the growth inhibition of Staphylococcus aureus would not have been possible without my facilitators, Mr. Robert Waters, Dr. Kyle Scully, Dr. Christopher Keller, and Dr. Nancy Carty. So to begin, I'd like to give a brief introduction of the organism we chose to study, and that's Staphylococcus aureus. So Staph aureus is a gram-positive bacteria, and you can see right up here, it looks like a cluster of grapes. Now, it is the most common cause of skin and soft tissue infections, two of those being cellulitis and folliculitis, also impetigo. So you can see an image right here of folliculitis, which is an infection of the hair follicles. So 30 out of 100 people are carriers of Staph aureus, so you may or may not be symptomatic. It does reside in the nares, which is the nostrils. And 2 out of 100 people carry the methicillin-resistant strain of Staph aureus. So because of its prevalence and its ability to acquire resistance, we thought it would be an important organism to investigate and to look into novel therapies for. So our next thing, why essential oils? So first, what are they? Essential oils are concentrated plant extracts, and they do come in 2 different varieties. So you can get a single pure extract, and those are something like cinnamon bark, and you do have to be aware that there are several different kinds of things that can come from cinnamon. In particular, you have cinnamon bark, you have cinnamon leaf. So even with the same plant, you can get a lot of variety, but also you have to keep in mind that there are different regions that you can get these things from, and so with each different region, a pure single extract can have a variety of organic components that go into it, and a lot of variety there. Our second type is the blend oil, which is essentially a blend of the pure single extracts, so put all together. And we were interested in looking into essential oils because they have gained popularity and they are now being used as natural therapeutic agents. So our experiment focused on thieves oil, and the reason we chose thieves oils is it has been used anecdotally to treat Staph aureus infections. And thieves oil does have cinnamon cassia bark, which we've done previous studies on all the pure single extracts that are in our lab, and we found that cinnamon cassia bark is very good at inhibiting the growth of Staph aureus. So we wanted to see if its function in a blend would have additive effects with the other essential oils in the blend, so to see if it would augment its ability to inhibit the growth of Staph aureus. So how did we do this? So we did a series of 3-day experiments, and our first day was the culture preparation day. So we took some of our strain BA977 and added it into a broth, let it incubate overnight, came back, standardized it to an optical density of 0.03 to 0.04. And what that essentially is telling you is that it tells you how many cells are in your solution. So we standardized that on day 1. On day 2, we took 100 microliters of our standardized solution and added it to a Mueller-Henton plate and distributed it evenly, so you can see the growth of bacteria right here. And then we added an antibiotic disc or a prepared essential oil disc. And so that's that white thing in the center. It's 6 millimeters wide. And the antibiotic disc came purchased, and the essential oil disc we prepared ourselves. It was a 6 millimeter disc, and we added 10 microliters of the essential oils that we wanted to test onto it. And then we put those plates in the incubator at 37 degrees overnight, and came back the next day and measured the zone of inhibition, which is this colony-free zone right here. And we took 3 measurements of the diameter and averaged that to get our ZOI, which is the zone of inhibition. So continuing on, here are our numbers for the antibiotics that are traditionally used to treat Staph aureus infection. So we got these antibiotics from the Infectious Disease Society of America's page for things that are commonly used to treat Staph aureus infections. and we wanted to test to see if it was resistant to penicillin as well as oxacillin. And so these are our numbers, and we took those numbers and compared it to the CSLI guidelines to see whether it was susceptible or resistant. And now we found that our particular strain of Staph aureus BA977 was susceptible to all of the antibiotics except for penicillin, to which it was resistant. So continuing on to our results for thieves oil, you can see here, so we have thieves oil at the very beginning, and these are the single pure extracts that were tested on their own. They're all components of thieves oil. And the ZOI is what we were looking at. So as you can see, thieves oil is quite low over here. And cinnamon cassia bark, which performed well as we expected. We did use statistics. We used a Kruskal-Wallis to compare all of the essential, the pure single extracts back to the thieves oil, and we followed it with a post hoc Bennett, so we didn't find any significance with that. But we did do a Mann-Whitney U to compare the cinnamon bark specifically back to the thieves oil, and we did find a significance of P less than 0.05. So something I'd like to highlight really quickly is how effective cinnamon bark is in inhibiting the growth of Staph aureus. And so I can tell you the average ZOI for cinnamon bark was 37.0 millimeters, which I'm just going to take you back to the antibiotics really quickly. And these are all ranked in order, and you can tell that, so clindamycin had the greatest zone of inhibition, and cinnamon cassia bark was 37, so cinnamon cassia bark is way up there. So that doesn't tell you anything about the therapeutic quality of cinnamon cassia bark, but it does tell you it's very good at inhibiting the growth of Staph aureus. So in conclusion, we would like to put out there that thieves oil is not an appropriate antimicrobial to treat Staph aureus. However, cinnamon cassia bark with its large ZOI was very promising. So it is the more active antimicrobial component in the blend. And because we did test it against all the other essential oils, our theory is that it was diluted by having the other essential oils in the solution. And so that's the reason thieves oil wasn't quite as effective. Now, future studies should focus on the cinnamon cassia bark oil, and we're going to be testing the MIC and the MVC on Staph aureus, the specific strain BA977. And thank you for your time. Any questions?
Just with the use of essential oils, so people, are probably spreading it around in their hands. Do you think the ZOI would decrease that if people are spreading the oil rather than just putting like a single drop in an area, which is how I know everybody has to test in the lab?
Mm-hmm.
Do you think that would decrease? I don't want to say it's efficacy, efficacy, but do you think possibly that if people were using it, they should use more of them?
So you have to really look into a lot of research on this, and I know for a lot of the essential oils that are effective at inhibiting the growth or killing the bacteria, that they're considered hot oils. And so if you put them directly on their skin, you're going to blister up, you're going to have a lot of reactions. So you want to be careful with that. I have looked at a couple of books where a naturopathic doctor suggests, here's the carrier oil you should put it with. If you are interested in using these oils as a therapeutic agent, I would actually recommend talking to a naturopathic doctor as far as how to use it and in what way you should use it. So, good question though.
I have a question. Do you think as essential oils become more popular as they have been, do you think there's any chance for resistance to start to happen with some of these?
Absolutely. So some of the people that are saying that essential oils, there's no really good website saying like use essential oils instead of antibiotics, but there are websites that are claiming that since essential oils have a lot of variation in their chemical composition, that bacteria have a harder time getting resistance to this. However, there are certain components that are the active components of the essential oil, so yes, I do think the more you use it, the more resistance you're going to get. So yes, definitely.
And why do you use Staph aureus?
So Staph aureus is a great gram-positive organism to work with. But again, because it's so prevalent and because of its ability to acquire resistance, we wanted to take a look at this organism.
Any other questions? Okay, thank you. Our next speaker, Gary, you're here, right? Yeah, okay.
So this is going to be our last talk for the session and for the symposium altogether. If you were a speaker during this session, in the back of your name tag you have a lunch ticket. So lunch will be provided in the cafe. If you were not a speaker, lunch is still in the cafe for purchase, so please take advantage of that. We also still have some of our posters upstairs as well as our exhibitor tables, so you can visit that during the lunch break as well. And we are going to try to have the student awards. We'll probably stick to that 1:20, maybe we'll do it a little bit earlier, and we will notify all of you that are here in the building. But we might do it just a little bit earlier than 1:20 since we are ahead of schedule. But if we do, we will let you know. So we are going to go ahead with our last speaker, Gary Vanderlaan, and he is from Gannon University and going to talk about non-pathogenic microbacterial models.
Cool, thanks. And I just want to let you know I'm going to be really fast, so I don't hold you from your lunch. And also the other thing I need to tell you is that I actually don't have any data whatsoever. So everything I want to show you is really a thought experiment that I think I might have something that might be a really good undergraduate research mentorship project. And so I'm looking for a little bit of feedback there too. So what is a diaminopimlet decarboxylase? This is an enzyme that basically makes lysine. Except lysine, like in humans, right? Lysine is totally essential. So we get it, meaning that we get it through our diet, right? But for a lot of organisms like plants and microbes, they have this enzyme, or they have something analogous to this, where they can actually synthesize it from scratch. The decarboxylase is just stripping off a carboxyl group. And so what they're basically doing is they're converting diamine pimlet directly into lysine by stripping off that carboxyl group. The operon that the very terminal enzyme is diamine pimlet decarboxylase, and it's encoded by the lyse A gene product in the lyse operon. And you know, for all of genus Mycobacterium, and there are many species actually in that genus. They all have this lysine operon, so that they have the capacity to treat lysine amino acid as non-essential. They can synthesize it. You can grow them in minimal media in the absence of all 20 amino acids, and they'll take a long time, but they'll still grow. they're totally capable of anabolizing everything for themselves. So anyway, so previous work, like going as far back as like 1955, have actually looked for drug blockers against diamine pimlet decarboxylase, because this is a really attractive enzyme, right? If it's non-essential to the microbe, but if lysine amino acid is non-essential to the microbe, but it's essential in humans, then in theory, if you had a drug that could block DAPDC, diamine pimlet decarboxylase, it shouldn't actually affect the host as much, right? Because we get all of our lysine through our diet. We don't actually have to synthesize it. So it should minimize some of your side effects there. And to date, like, and the reaction basically, I forgot to mention, the diamine pimlet substrate actually has two stereocenters. So there's a dextro and a level for the natural substrate here. And so the lyse A gene product is the diamine pimlet decarboxylase, and you're just stripping off a carbon dioxide here so that you can turn DAP substrate straight into lysine amino acid. And this is happening in a lot of plants and every species of genus Mycobacterium. Okay? And these are just some of the key amino acid residues to run the active site, essentially, in the center right here. So there are two known blockers against DAPDC, and these are tested in vitro. So we have, and these are both working by competitive inhibition. And so you have lanthanine sulfoxides. This one's probably the earliest one. They both basically compete for active site access, right, on the enzyme with the dextro level form. This is the natural substrate. And so, you can, with like 2 isocenters, right, stereocenters, you can actually make like 4 stereoisomers, right? And so you could have like a DD form, a DL form, an LD form, and an LL form. And this particular one is, of all the synthetic stereoisomers, is the strongest one in terms of its competitive inhibitory role. So, a real simple assay might be to maybe just look for zones of inhibition and look for synergy, right? Like if you were to put if we were to pick like, like tuberculosidos or tuberculostatics that had antimicrobial properties against genus Mycobacterium, maybe we might do something like the Kirby-Bauer test, right? And we might do like a disc diffusion assay. And so here, like I might take a Petri dish, and this is all M5 minimal media, so it's lacking all the amino acids, right? So it's going to take a while for these microbes to grow. But before we drop these discs on here, we're going to just make a whole lawn. We're just going to inoculate the whole thing with Mycobacterium smegmatis, the non-pathogenic form, because I don't want to kill my Gannon students. But anyway, what we might choose is we might actually choose like discs that might block mycolic acid synthesis. Mycolic acid is a key wall component of genus Mycobacterium. We might pick something that's like going to block a transcription, like the beta subunit of RNA polymerase in prokaryotes is this RPOB polypeptide. And if these guys actually synergize because of different mechanisms of action, we might see, like when we're looking for the zones of inhibition, we might see like them grow towards each other, so to speak. So you might see something like this. So I kind of exaggerated it a little bit, but you might actually see clear zones right here that approach each other if they actually do synergize at those specific concentrations. And the idea here is, like, will we see any synergism with either lanthanine sulfoxide or with like one of the stereoisomer competitive inhibitors as well, right? And the reason why this is actually really important is we're actually running out of drugs actually to fight like Mycobacterium tuberculosis. And so these are really common drugs that are used like in the clinic against Mycobacterium tuberculosis, but there are like multi-drug resistant forms where they're resistant to one of these two, and there are XDR forms, like extreme drug resistant forms, where they're resistant to more than one essentially. And so if we can actually show that this might be effective, we might be able to add another antibiotic or something like that to the mix. And the sort of bigger picture that I was kind of imagining here, this might be kind of fun, because everything in genus Mycobacterium loves to live inside monocytes, right? Like Mycobacterium loves to actually invade monocytes, like alveolar macrophages are good targets. And so what might be kind of cool, right? It is just kind of a crazy idea, but maybe if we had like, if you had like a piglo expressing Mycobacterium smegmatis, right? And you can get it inside like a monocyte, like a THP1 monocyte. And maybe you can get that monocyte to glow like red. So you're doing this all in vivo, right, with transfection and transformation. One of the cool things about M. smegmatis is, unlike M. tuberculosis, whenever the pathogen actually gets inside the monocyte, these are actually very good at evading the phagolysosome pathway, so they don't get degraded, and they can actually hitchhike inside the cytosol of the macrophage. When the macrophage finds out that the organism is living inside the cytosol, the normal response is to execute apoptosis. And that does happen with M. smegmatis infection. And so what might be interesting is if we have like a DAPDC blocker or something like that, and we might be able to ask whether or not they're able to actually receive lysine nutrition just from their host environment. If we were to block, if we had, for example, like Mycobacterium smegmatis infection of THP1 cells, and then we might do that in the context of like lanthanine sulfoxide or something like that, and see whether or not they can actually still induce apoptosis of their host cell or something like that. But this is kind of like the direction I'm thinking about. But I got to build my tools first. This is my first semester here. Anyway, I don't want to hold you guys from lunch or anything, but if you guys have, if you're interested in something like this, or if anybody's actually working on tuberculosis, we're not qualified. We don't have the biosafety lab at Gannon, the rating at Gannon to do tuberculosis research. But if anyone's interested in this, we can swap out M. smegmatis for tuberculosis and do it in like a clinical setting. I would love to talk to you. Okay. Thank you, guys.
Thank you. Does anybody have any questions? Chickens. Well, I do encourage, since we are going into lunch break, if anybody was interested in discussing, please see Gary after or during the lunch break. All right, so we are going to break for lunch. The time right